our mission is pretty audacious.
It is.
We are going right inside the cell's nucleus, pulling back the curtain on the deepest secrets of the genetic code.
Yeah.
We're going to be tracking this massive technological leap that took us from just sort of rudimentary observation to the blazing speed of modern genomic computation.
It's an incredible story of acceleration, really. When we talk about how we started reading DNA, I think the best way to frame it is as a kind of technical despiraling of genetic knowledge.
Spiraling.
Exactly. We had the blueprint, that famous helical coil locked away in the nucleus.
Right.
But the science we're diving into today is all about how quickly we develop the tool, the chemistry, the physics, and the computation to read that code faster. And
to really understand the magnitude of the shift, we have to start at the absolute beginning, the foundations. We don't need to dwell on the basics, but it's pretty humbling to reflect on Gregor Mendel. He was the original source of insight here.
It really was.
He essentially stumbled onto the whole concept of inheritance, of genetic encoding, not through some billion-dollar lab, but just through simple observation and methodical recording.
And what's fascinating is that methodical recording, it grounded the entire field. He established the concept of an instruction set.
Okay.
But the molecular structure itself, that was the next great barrier. Once the structure of DNA was finally resolved, scientists realized that the structure contains the genetic information. And critically, this structure is copied continuously for what the sources call all genetical toil.
Which means what the constant effort required to just replicate life?
Exactly. And that code is standardized. It relies on just 4 chemical components, the standard bases for each pair.
Right. A, T, C, and G.
And understanding the consistent universal structure is what allowed engineers and chemists to start building machines to read it. That basic molecular structure is the target for every single piece of sequencing technology that came after.
Which brings us to the first major hurdle. You have this incredibly long, complex sequence of bases, and the earliest attempts to read it were, well, they sound agonizing.
Oh, they were. If you think of Frederick Sanger's initial methods, I mean, they were pioneering, absolutely groundbreaking for their time.
That's so slow.
But truly a basic slow manual mode. They involved these meticulous step-by-step chemical reactions that were just so time-consuming, and they required massive amounts of starting material.
So reading even a tiny piece of a genome was a huge task.
A monumental task. It was completely unsuitable for reading, human-scale information.
So how do we make that jump from this painstakingly slow manual labor to the massive automation that really defined the turn of the century?
Well, this is what we call the first sequencing revolution. It's often called next-generation sequencing.
Okay.
And it wasn't really a mechanical leap. It was a chemical one. It was driven by what the sources describe as polymerase chemical evolution. And that innovation fundamentally changed how we prepared and read the DNA.
Let's get into the mechanics of that, because that feels like the key technical insight. What did that chemical evolution actually enable?
It enabled massive parallel sequencing. So instead of trying to read one long fragment manually,
Which is the old way.
the old way, yeah, they could shatter the entire genome into millions of tiny fragments, attach these fluorescent chemical tags to each base, A, T, C, or G, and then read them all at the same time.
So the DNA isn't being read by a pair of human eyes or manual chemistry anymore.
Not at all. The mechanism relied very heavily on base illumination, coupled with increased laser detection.
Base illumination, so lighting them up.
Think of it like this. Each base lights up a specific color under a powerful laser. The computer snaps a photo, determines the color, and therefore the base, at millions of locations simultaneously.
And that automation is what drove the high speed and reduced pricing.
That's what made the $1,000 genome a reality. It completely revolutionized biology and, of course, medicine.
That is such a fundamental shift. You go from manual chemistry to automated optical reading.
The next big breakthrough needed to address a couple of problems. Complexity and the time it took to prepare the sample.
Right.
And this leap, it was heavily driven by what the source calls a very clear commercial intention, primarily by companies like Oxford Nanopore.
This is where we get to that famous invention, the nanopore electrophoresis passage. For listeners who follow this topic, I mean, that phrase is central. But what exactly is a nanopore? How does it read DNA differently?
Well, what's so fascinating here is that the principle is physical. It's not primarily optical or chemical like the last generation.
Okay, physical.
Imagine a membrane with a microscopic hole in it, like a biological toll booth, just a few nanometers wide. That's the nanopore. And you have a constant electric current flowing through this hole.
A microscopic hole with electricity flowing through it. Got it.
Now, you feed a single strand of DNA through that pore. This is the breakthrough process that garnered so much fame.
A single strand.
A single strand. And as each base, A, T, C, or G, passes through, it physically disrupts the electric current in a uniquely measurable way.
So each base has its own electrical signature.
Precisely. A detector instantly registers that subtle signature, and a computer translates it directly into the base sequence in real time.
That is just a staggering jump in scale. I mean, we've gone from massive parallel batch sequencing, which took hours or days of prep, down to reading a single unamplified molecule as it just slides through a tiny hole. The sample prep must be minimal.
It is. And the result is instantaneous. It's also the ultimate form factor innovation. Nanopore allowed sequencing devices to shrink down to the size of, like, a thumb drive.
Making genomics mobile.
Mobile and immediate.
Yeah.
And that accessibility is just as important as the speed.
Okay, so sequencing the code is one thing. But once you have that digital sequence, you need to know what the molecular machinery is actually doing.
Exactly.
So that means shifting from reading the code to, I guess, visualizing the actual physical structures in action.
That's the next pivot. We have to now tackle the challenge of seeing the unseen. And traditional light microscopes, the ones we've used for centuries, they're basically useless here.
Why is that?
Because the molecules we're studying are smaller than the wavelength of visible light. The light just scatters. You see nothing clear.
So if light doesn't work, what does this new technology use?
It uses beams of electrons instead of photons. We fire these electrons probe at the frozen molecules.
Okay.
And the signal from these electrons is then captured and processed computationally to form what the sources poetically call a display ruse.
Let's unpack that phrase, display ruse. If it's not a true visual image, like from a camera, what data are the scientists actually recording?
Well, they're gathering all this complex two-dimensional projection data from thousands of different angles.
Right.
And then they use algorithms to mathematically reconstruct the three-dimensional shape of the molecule. The critical result here is that this process allows for visualizing the motion in the three dimensions of these incredibly complex chemical convoluted molecular visions.
That connects the data perfectly. So now you don't just have a static sequence of A's, T's, C's, and G's.
No.
You can actually watch the physical DNA or the RNA or the proteins, the machines that execute the code as they fold and move and interact in real time.
And this is where the human expertise and massive computing power become indispensable. We rely on what the sources call the artful, clever clogs.
A clever clog.
The computational biologists, the bioinformaticians, and the AI systems who have emerged on sites dedicated entirely to processing this genomic avalanche.
And what are these sophisticated systems actually doing with all that data? It's not just, basic data storage.
Oh, far from it. These clever clogs, they are hungry to swallow and compute myriads of bytes from the increasingly rich genomic menu and its mutations.
So they're not just reading the code?
They're not just reading it. They are comparing it across millions of individuals, identifying subtle mutations, and then modeling the potential effects of those mutations on the 3D molecular structures we just talked about.
So they connect the sequence to the structure and the structure to the disease.
Exactly. That speed of comparison is key. Before, finding a single relevant mutation might take months of effort. Now it's done in hours.
And this leads us to why all these technical breakthroughs, from base illumination to nanopores to 3D visualization, why it all ultimately matters to you, the listener. What is the ultimate tangible application?
It's personalized preemptive medicine. The ultimate goal of this entire journey is using this highly contextualized knowledge to find, edit, and resolve rare medical situations.
I see it.
The ability to sequence a patient's genome quickly, see the mutation, and then visualize how that mutation impacts the physical molecular machinery in 3D, it means we can design targeted therapies that were simply impossible when genomics was still in that slow manual mode.
It shifted medical research from a very broad brush approach to highly specific molecule-level intervention.
That's the power of the computation matching the speed of the sequencing.
So if we synthesize this whole process for you, the listener, we have tracked this journey from Mendel's notebook, that original observation of traits, through the chemical revolution of parallel sequencing to the lightning-fast single-strand nanopore technology, all the way to electrons probing molecular visions and computers processing myriads of bytes of data, all in the quest for health.